I'm Dr. Michael Girardi from the Yale School of Medicine, Department of Dermatology. And I'm also a physician scientist here at Yale. So I'm going to talk to you about basic science research for cutaneous T-cell lymphoma, advancing our understanding and treatment for this disease. So being at the Yale School of Medicine, where we do a lot of our basic science research, connected to the hospital where we treat a lot of patients, including in our comprehensive cancer center, we have a lot of physicians and scientists working together to try to better understand this disease, why it happens, what happens to patients, how we can predict what will happen to patients, and how to better treat them. So we have a lot of systems working together. We have a lot of physicians and scientists working together. This type of research, called basic science, is about what we do in the lab, as opposed to what we do and offer to patients with clinical trials. Dr. Foss and Dr. Sethi will be talking about that aspect of treatments that can be offered to patients as a type of research. Basic science, we're trying to address some of the current challenges in CTCL. There's a difficulty in early diagnosis due to lack of reliable markers. There's variability in disease progression and outcomes. How can we understand and predict what will happen to certain patients? And though we develop and have seen new treatments come online for our patients, there's still a limited number of treatment options for advanced stages. So working together in the laboratory, we try to figure out ways to attack some of these challenges. Here's my lab at Yale and some of the things that we've been involved with in research in trying to understand this disease. We've been looking at the genetic changes that occur within patients. We've been looking at the immune changes that occur within patients and how we can harness the immune system to better attack the cancer cells. There are microenvironment studies, including bacteria that's on the skin that might promote the cancer. And then we have studied some drug discovery and development opportunities to try to help develop new therapeutics for patients. So let's talk about genetic and molecular studies. One of the ways that we can look at helping to understand this disease is to identify driver mutations and potential therapeutic targets. So by sequencing the DNA of the cancer cells across many patients, we can see that, yes, there's a diversity. Not every patient is the same, but there's also some commonalities, and these might serve as new targets for treatments. We want to understand the disease mechanisms at the cellular level. How do the abnormal cells communicate with your normal cells in the body? For example, CTCL cells will affect your normal immunity and suppress it so that you might be more prone to infections in advanced diseases. And we want to develop new diagnostic markers. That is, new ways of identifying DNA or RNA or proteins that'll tell us that this is definitely CTCL and which subtype. CTCL is genetically diverse, and again, we want to find common targets. This represents a very complex diagram of some of the proteins that communicate in cancerous CTCL cells. And these are some of the targets that we've helped identify for treatments over a series of publications that are available to the scientific community. In immunology research, we are concerned about immune system dysfunction in CTCL, how the CTCL cells cause havoc among the normal immune system. We are also concerned about how the T cells drive itch, such an incredibly challenging part that affects so many patients with CTCL. And developing immunotherapy approaches, meaning how we can re-harness the immune system to attack the cancer cells. Here's one of the ways my lab has been working to develop an immunotherapy for CTCL. Here we have a patient whose CTCL has a marker of a T cell receptor V beta 2 on it. And what we'd like to do is try to eliminate CTCLs by targeting that very specific marker and leaving the normal T cells alone. In this example, we see an engineered T cell that will target that TCR V beta 2 and eliminate it, thereby leaving the normal T cells intact. So to eliminate the cancer cells by minimally affecting the normal immune system is one of the big goals of developing new CTCL treatments, as an example. Now, this is early on, not yet available, but we're still working on it in the lab. Another aspect of basic science research, which is very exciting, is high throughput techniques and artificial intelligence. So all this information that, for example, comes from sequencing DNA from patients' cancerous cells in the skin or in the blood is really ripe for rapid production of a lot of data points. We can even do a lot of this sequencing on a single cell basis. So we can see within the same CTCL patient how there may be some changes that happen to the cancerous cells over time. We can also understand the diversity that happens at a given time. All this data can get fed into very advanced, high-functioning artificial intelligence and machine learning programs, and of course work with a lot of colleagues in computer science and data science who are on the advanced edge of this type of research to identify, again, keys to understanding what's different about each patient, what's likely to happen to that patient, and what might be the best treatment for a particular patient. So very exciting research in high throughput and artificial intelligence. So after going over some of the areas where basic science tries to focus on CTCL, I want to talk about why your participation in these matters. Again, nothing is really done to you when we talk about basic science research. This is about you providing access to your skin or blood, for example, samples of such that will allow scientists to try to decipher some of these challenges in CTCL. So you as a patient can help accelerate research progress for yourself and others with CTCL. You can help develop personalized treatment approaches because the more different patients that participate, the more diverse information we can get about the disease in each of so many patients. And this will contribute to improving diagnosis and prognosis for future patients. So you're helping to leave a mark on this disease for many years to come. So how do you help? So you need to sign permission. It needs to be explained to you what you're actually offering and what's going on. But typically, we're talking about skin biopsies and blood draw samples. Not typically anything different than what you've had to treat your disease and diagnose your disease, but these samples will be used for research. Sometimes you might be asked to share your personal health data. That is not your personal information, not your name, and not anything about you personally, but only the data that is about your disease. So it's what we call de-identified. And then participation in clinical trials and observation studies are something that you can also be part of that's going to be talked about later by Drs. Foss and Dr. Sethi. That is a different type of research. Those are clinical trials that can help bring a new treatment directly to you today. So in summary, the impact of your contribution of basic science can help advance our understanding of disease mechanisms, how it happens, why it happens, development of new therapies and diagnostic tools to help everyone with CTCL, and ways to improve the quality of life for CTCL patients. So basic science is crucial for advancing CTCL treatment. Your participation can make an absolute significant difference. And together, physicians and their patients, and the research laboratory personnel, like you see here in my lab, can work towards better outcomes for CTCL patients.